So today we're moving on to modern classification. Thinking back and looking back to Monday's work, we talked about Carl Linnaeus, who developed the binomial nomenclature and his system of classifying organisms. Linnaeus classified according to visible similarities and different traits in adult and mature organisms. So we had to wait for organisms to fully mature or become adults before they would, compare and contrast one organisms to another. What happened with this is that it had limitations. So if we look here at the picture below, we have the dolphins here in the middle. Just visually looking at the dolphins and the fish and then the man in the bottom right, dolphins look visually much more like a fish than the man, okay? But we know that dolphins are mammals. They have mammary glands. They care for their young. They have hair on their body, okay? So they are much more closely related to the man, okay? But if we just strictly use Linnaeus's categorization or classification, we would look at as dolphin as a fish, and we know better than that. Moving on, so what we do is we use phylogeny, and phylogeny is a representation of organisms based on describing evolutionary relationships, okay? So what we are doing is we are taking Linnaeus's hierarchy and just adding the element of time to it, okay? So evolutionary is just the change of these relationships over a period of time, okay? Again, all we're doing is we're taking Linnaeus's hierarchy and adding a period of time to it and how it has changed. Biologists now group organisms into categories that represent lines of evolutionary descent or phylogeny, not just physical similarities. So again, we're looking at a timeline along with the classifications. It's called the evolutionary classification model. Again, evolution is just change over time. So we're looking at how organisms have changed over time. How have they become more similar? How have they become more different? How has, one organism or two organisms started off very much like a common ancestor and now they've branched out? How have we changed over the time? In the evolutionary classification model, all members of the genus classification share a most recent common ancestor. This would be like you and your brother, or you and your sister, you and your siblings share your parents as a common ancestor. Now your parents are maybe, 40s, 50s. They are your most recent common ancestor. All members of a family share a common ancestor a little further back, who's a little bit older. This would be like you and your cousins share the same grandparent. That grandparent's maybe in their 70s, 80s, 90s. They're a little bit older or further back in the time of common ancestor to you and your cousins. We can keep going. The members of the order would be like great grandparents, etc. The higher the level of taxon or classification, the further back in time the common ancestor of all organisms is found. Like I said, it's still Linnaeus's hierarchy. Now we're just adding a time to each level. An example, if we look at these two drawings, if we look at the one on the left first, get my pen here. We have a barnacle and a limpet, okay? They look very similar. If you went out to Maine or somewhere along the sea and you grabbed a barnacle and a limpet, you probably wouldn't be able to tell the difference, okay? But if you had a crab and a barnacle, you would be able to tell the difference. One has a shell, outer shell, maybe a bilayer or bishell. One has appendages. You can see the very different reactions. Okay, if we were sticking with Linnaeus's classification, you would classify barnacles and limpets together. Okay, but what we've seen with the limitations is we're going to add another part of time into the classifications, and we're actually going to look at the time of the life cycle. Okay, so over here on the right is the evolutionary classification. Okay, we call them a cladogram. And if we look at tiny free swimming larvae, we are looking at a larvae of these organisms. Okay, so instead of just looking at the adults, we're now looking at an immature larvae. To the left, we have segmentations. We molt our exoskeleton or external skeleton, and then you can see the branch to the barnacle and the crab. The limpet is off here on the right under a mollusk, and then the crustaceans are over here. If we switch pages here, you can now see that a barnacle at one time has appendages. Okay, if we look at the life cycle of the crab here, we go eggs, zoea, and then a megalupa. Okay, again, appendages very similar to the barnacle. We now know that at some time, again, they must have shared a common ancestor where developing into this image or developing into this organism included this step. Somewhere along the lines, the crab decided to keep the appendages, and then the barnacle decided, you know, we decided not, it's best for us to just go about it this way. These characteristics that, you know, make us determine one organism from the next are derived characteristics, and we'll talk about here. To refine and even kind of, you know, narrow down the evolutionary classification, they use cladistic analysis is used. And again, this is just doing the analysis of cladostatistics, which is just, again, looking at time. The derived characteristics are characteristics that appear in recent parts of lineage, but not in the older part. This is shown through a cladogram, and here's an image of one. So we have chordates, which humans are a part of, and you can see as we go down to the left here, we go further back in time, and we have kind of less important things that go along with them. So if we move down all the way to here, organisms at this time all had columns, which mean they had kind of one way in, one way out. Okay? At this time, it is one way. All these organisms up here have one way in and one way out. Okay, goes in and out, not in and out the same way. If we keep going back, the three germ layers, development, okay, and bilateral symmetry. If we go back to humans and we talked about stem cells, you guys talked about how the stem cells form into 3 layers, right? That happens all the way back here. And then the really only relationship we have to sponges would be that we are multicellular. Lastly, there's another couple of classifications that we can use. One being genes. We can look at genes that are shared between organisms. And then lastly, DNA. DNA is very much how they determine paternity suits, you know, who's the daddy, if you will. They can also, you know, determine long lost relatives. They can determine a lot by looking at the DNA and exactly how similar it is to other people. Okay. Hopefully you learned a little bit about modern classification and how we still use Linnaeus's classifying, only we've kind of upped the ante and we've added timing into it. And now we take not only how similar organisms are, but at what time they have branched off from a common ancestor.